So for the next 15 minutes or so, I'll just briefly describe to you something about data. The data I'm going to present to you mainly come from a recent study led by Ron and supported by Linda as part of the ME CFS project. So when we talk about big data, as we heard today and all the brilliant talks in the past 2 days, there are basically two types of big data in medicine. One is that for a popular a common disease like ME CFS, one approach would be if we have sufficient support, perfect, we can actually try to examine what's wrong in every patient around the world. And as I hold this ribbon here, it says there's 20 million patients around the world suffering this disease. So one type of big data with sufficient support would be to look at each one of them and compare their health record and molecular signature with other common diseases. That would be the ideal situation. But since we can't afford to do that at this moment, the alternative approach that started about a year ago was to carefully select a few severely ill patients and try to do as much as possible, basically turning every stone that we can afford from the gene level, the protein level, the metabolites level, as we have heard so far from multiple studies, as well as cells, tissues, and behavior of the patients, together with the microbiomes and the environmental exposure of these patients, and try to understand, what would be the abnormal signature for this disease. And perhaps a longer-term goal is, can we actually change some of the factors and be able to treat the patients? So more specifically, this is a list, actually a partial list of the tests that have been conducted so far in Ron's team. And I won't go through all this, but because of time. But if there's interest, we can follow up offline. And I guess part of the important goal of this study is to make the data publicly available, perhaps before publication, to the entire research community. So people who's interested in this disease process can actually take a look at the data and help their own research. So in terms of analysis, since I'm a data person, in terms of analysis, there's obviously 3 types of things that we typically do. One is to look at individual patient across different dimensions of data and try to understand what could one factor affect the abnormalities of another factor at another level. For example, genetic factor affect metabolite level, for example. The second one, which commonly being conducted, would be to compare patients as a group with controls. So that's the one in the middle. And the third one would be, can we actually take what we learned at each individual level and put all the things together? That's obviously the ultimate goal. So this is sort of the baseline information. I think in this room, everybody must have already been familiarized with this data. But basically, this is the set of quality of life parameters using SF-36 in the patients that we selected in the study, comparing to large surveys using SF-36 to evaluate the quality of life of other chronic diseases, from heart failure to chronic lung disease, arthritis, ischemic heart failure, diabetes, et cetera, et cetera. And on the far left side, or your right side are those controls. So you can see that the quality of life of CFS patients is unfortunately much lower than some of the common other diseases. And as Dr. Hansen already mentioned earlier today, the factors that are actually less affected are those ones that relate to emotional health and mental health. So those are the ones that are actually not affected that much in CFS patients. If we monitor the behavior of these patients, as you can see here, this is just the simplest example, the number of steps that the patients take on the red ones are the controls, and the blue ones down there are the patients. So we can see that patients are heavily limited by this unfortunate disease. And the dotted lines in the middle are the average values of the steps that people in the US take from 2 separate large-scale studies. So we know that the CFS patients suffer from severe debilitating disease, and this is just one presentation. If we actually look into their electronic health record, the frequencies of the symptoms or complaints that they have, and I think we owe the research community, owe the patients a solution to this problem. And as we have heard today and also over the past 2 days, we're trying our best to figure out the underlying problems. So before I give you some of the examples of the data, let me just, since I'm a computational person, let me just caution that these are the preliminary results, that we feel confident that the results are true in our study of a small set of patients who are severely ill. It's not known whether the findings that I'm about to show you would be able to translate to other patients. So that's the caution. This first data set is a set of about 250 clinical lab tests between patients and the controls. And out of the 207 tests, about 27 of them are significantly different between patients and controls. Because of time, I'm only showing you one, which is the cortisol. So this basically shows that the cortisol level of the patients right after they wake up is significantly lower than the controls. But as time goes on, the level even out. So it seems like there's something different between patients and the controls in their sleep. I'm sorry, I'll just quickly go over all the different type of data we have at the moment, so it's quite brief. So we also look at 61 cytokines in the blood of the patients versus controls, and 21 out of the 61 are statistically significant. These two shown here on the left side is leptin, which is known to be different between patients and controls. It's significantly higher. However, you can see that some of the patients, for example, this one, would have much lower leptin comparing to controls. So that reflects this heterogeneity in the patients. And on the right side is this brain-derived neurotrophic factor, which is known to indicate the activity of the central nervous system. And we can see that across the board in patients is significantly lower than in controls in this set of severely severely ill patients. And we also did the metabolome analysis, and As mentioned, similar to what's mentioned earlier today, there are significant number of metabolites that different between patients and controls. And again, I'm just showing two as examples. On the left side is this indopropionate, which is much lower in patients compared to controls. And on the right side is lysine, which we know is one of the amino acids, common amino acids, that's higher in patients compared to controls. Some of us probably recognize that this indopropionate is a known neuroprotectant agent, and it's involved in tryptophan metabolism. Some of the researchers in this room actually are doing some studies on tryptophan metabolism and see whether it's indeed impaired in patients versus controls. We also know that, for example, for the indopropionate, it's actually mainly produced by gut bacteria. So if we actually look at the gut bacteria between patients and controls, we can see that the Firmicutes, which shown in green up there, between patients and controls, and the Bacteroidetes, which are gram-negative bacteria. So the Firmicutes are gram-positive bacteria. The Bacteroidetes are gram-negative bacteria, and these are sort of common ones in our gut microbiome, and they're different in terms of ratios between patients and controls. And perhaps more strikingly, you can see that these viral microbes are mainly dominant in the patients, but in selected patients. So what's going on in their gut might contribute to some of the phenotypical differences in patients. And I think there were previous publications about it already. If we just look at the microbiome diversity in the gut, this basically shows the diversity, and we can see that there's a huge heterogeneity between the individual patients. So some patients have much higher diversity. There are a lot more different type of gut bacteria in their gut, and some of them, however, have much lower diversity. And this just shows the whole genome sequencing data of the patients. If we just put a limit on the variance to be larger than 25% allele frequency in these patients, we can actually find a large number of the variants. Obviously, this is similar to a lot more of different studies of diseases. The key is to identify a subset of this that might be useful for, might be causal for the disease, and that's what we're trying to accomplish. As I mentioned earlier, one of the important goals of this study is to get the data ready for the scientific community, because we have to work together to have enough number of patients studied to come up with definitive conclusions. I think we're roughly 95% done, and hopefully that can be released very soon, so we can show not only the data but also the results through this website. So about the next steps, one is, or the current steps we're working on in the lab. One is to compare the signature of ME CFS patients with other diseases. This just shows one of the studies only looking at the gene expression, and as you can see, at least to the surprise of me, the most similar pattern, at least in the few hundred patients we look at so far, in gene expression, the most similar one is actually inflammatory response. And we can also ask the question across different type of analysis. What, for example, are the measurements that would be most correlated with, for example, vitality, energy, and fatigue report in the patients. And this would be the subset of measurements that would be most correlated, and then we can dig in further, whether any of this can be perturbed, for example, or whether any of this can be looked further in this disease study. We talked about the indopropionate that's just used as a contract that does not seem to be correlated with the degree of fatigue, at least in the patients that we studied. The other thing that's going on, this is the last slide, in the lab, is to actually try to understand the causal effect, because fatigue, to some extent, is a problem in metabolism. So we're actually trying to use math to model, as you can see on the left side, the molecular details of the metabolic system in our body, and use the data that I just described to you to understand perhaps there's bottleneck somewhere in the metabolic system. And I hope I'll be able to report to you the next time what we find there. And with that, I'd just like to thank to the team, the computational people who actually did the work, and especially the NME CFS research group, who actually really did the heavy lifting of carrying the patients, recruit patients, collecting samples, generating the data. So as computational person, I think I'm really in debt to all the researchers in this room, and I hope we have a chance to collaborate in the future. Thank you.